morning. My name is Paul Hansen. I'm a corporate fellow at the Oak Ridge National Laboratory, and I'd like to welcome you to the virtual tour of the spruce and peatland responses under changing environments experiments that we simply call SPRUCE. SPRUCE is an experiment to assess the response of northern peatlands, a high carbon ecosystem, to whole ecosystem increases in both temperature and exposures to elevated atmospheric CO2 concentrations. The SPRUCE experiment is funded by the U.S. Department of Energy's Biological Environmental Research Program within the Office of Science at the DOE. It's also a cooperative venture between Oak Ridge National Lab and the USDA Forest Service with interested university cooperators. SPRUCE is located in northern Minnesota in Itasca County at the very southern edge of the boreal peatlands of North America. SPRUCE is a large-scale, largely automated manipulation with 10 warmed enclosures. The treatments are randomly deployed in a regression design, which includes 5 temperature treatments plus 2 measured ambient plots. The temperature treatments are no energy added, but fully constructed enclosures, temperatures that are raised 2.25 degrees Celsius above the ambient reference background of the no energy added enclosure, and 3 other warming treatments of plus 4.5, plus 6.75, and plus 9 degrees C above the ambient or non-heated enclosure. Half of those 10 plots, there's 2 for each temperature treatment, receive elevated CO2. They receive elevated CO2 because we anticipate that future warmer climates will be associated with a higher atmospheric CO2 concentration, that's carbon dioxide, that would lead to potential beneficial effects on plant growth. We do use a whole ecosystem approach. That was the goal of the SPRUCE study, was to build an infrastructure that could heat the system from the tops of the trees all the way down to the deep within the peat profile. The key SPRUCE science questions are: will deep below ground warming in the future release 10,000 years of accumulated carbon from peatlands that store approximately 1/3 of the Earth's terrestrial carbon? And at what rate would those warming treatments release the carbon? We're also interested to know whether the release of carbon will be in the form of carbon dioxide or methane gas. Because we've built all of this structure, we can also ask additional questions about ecosystem responses. Are the peatland ecosystems and organisms vulnerable to atmospheric and climate change? And what changes in those organisms and this ecosystem are likely? And at the full scale of the intact plots, will ecosystem services, for example, regional water balance, be compromised or enhanced by atmospheric and climate change? So as we proceed over our 10-year duration of our study, we're looking for what is the pattern of responses to trees versus shrubs versus the sphagnum moss versus microbial communities below ground to this wide range of warming treatments.
My name is Robert Nettles. I'm the operations team lead here at the Spruce Project in northern Minnesota. Our team has been tasked with accurately and consistently delivering the environmental manipulation treatments. 10 octagonal open-top chamber greenhouse structures were constructed in the bog that allow us to execute these manipulations all year round. Heating is performed all year. CO2 additions are only performed during the growing season. These enclosures were dropped directly down into the bog. No plant species were added, no plant species were subtracted. Each enclosure is approximately 15 meters in diameter and 8 meters tall. The open top allows for natural air exchange and also for natural precipitation infiltration. The first treatment that we're going to talk about is deep peat heating. Deep peat heating in these plots is accomplished by employing 67 100-watt electrical resistance heaters that have been pushed into the bog. There are 3 concentric zones that they have been installed in, and each of those is controlled with a constant feedback loop, PID control. The second treatment that we're talking about is atmospheric warming. Atmospheric warming is accomplished by employing 4 different indirectly fired propane air handlers per each enclosure, and it's also controlled by PID and constant feedback. The third treatment that we talk about is atmospheric CO2 additions. CO2, as was mentioned previously, is only deployed during the growing season when the foliage can actually take it up. Pure CO2 is injected into each air handler duct and is then mixed with ambient air and administered to the plot. Once again, also a constant feedback loop, PID control. All of the enriched plots receive 500 parts per million of carbon dioxide above ambient conditions. Maintaining environmental manipulation targets for a project of this size requires significant resources. We generally use on the order of 4,000 pounds of liquid carbon dioxide a day when we're fumigating. Electrical usage is generally about 8,500 kilowatt hours per month. And on a weekly basis, we use approximately 5,000 gallons of liquid propane fuel.
One of the things we're trying to do with the SPRUCE experiment is to determine the extent to which the warming treatments with and without elevated CO2 can lead to changes in net carbon exchange from this whole ecosystem peatland, and determine if in fact the system is gaining or losing carbon. The way we're doing that in the spruce peatland is taking a series of observations. We measure above ground tree production, above ground net primary production, through a combination of terrestrial LiDAR scanning of the canopy tree heights and the canopy volumes, and annual wintertime measurements of the diameter growth of the trees. We also measure production, ANPP, of the shrub layer above ground. Then we have a group that's looking at sphagnum production, and they isolate individual bundles of sphagnum in the bog, check whether or not they have gotten longer, that is, whether they've grown during an annual cycle, and then they also determine whether the sphagnum continues to exist, that is, we are seeing some mortality. And then we have a group looking at below ground net primary production through a series of harvested root cores. In addition to those 4 different measures of net production from the autotrophic organisms of the ecosystem, we measure whole ecosystem losses of CO2 and methane flux from some large collars that are embedded in each of the chambers. Measuring those collars periodically throughout the growing season allows us to interpolate the total amount of CO2 and methane flux leaving the system on a diurnal basis or throughout the year. And then we can subtract those carbon losses from the production losses from the 4 measurements of production that I just described to yield net carbon gain or loss from the ecosystem as a whole. What we are seeing in that context is a linear decline or a linear loss rate of carbon into the atmosphere. The net loss rates are on the order of 30 grams of carbon per degree C of warming every year. That rate of warming is indicative of a loss rate that's 4 to almost 20 times greater than the rate at which the peatland carbon has accumulated over time.
I'm Colleen Iverson. I am a senior scientist here at Oak Ridge National Laboratory, specifically interested in the interface between plants and the soil beneath our feet. So one of the things that we're interested in is how plant roots respond to warming and elevated CO2. And the roots are important for acquiring resources for the plant, but they're also an important component of the carbon cycle. And well, these roots, these fine roots, only live for a span of a year. And so when they die and decay, they become part of the peat around them. We want to understand when they're born, how long they live, and when they die and start to decompose. And we want to understand where that happens in the peat. And so the way that we measure what's happening below ground with roots is we use mini rhizotron cameras that are inserted into a clear tube that we bury below ground at an angle. And we in turn also have these automated mini rhizotron cameras where we have buried mini rhizotron tubes with a little robot inside of them that carries around a camera and takes thousands and thousands of pictures every week and sends them back to us. And the cool thing about the automated mini rhizotrons is that these images are at 100x magnification. And so we can see the fungal partners that associate with the plant roots and help them to acquire water and resources for the plant. So in terms of the findings that we have, so what are these roots doing in response to warming and elevated CO2? And what Dr. Avni Malhotra has found is that for every degree increase in soil temperature, there's a 130% increase in shrub root length. And so what we've seen in other places with drying is that these ericaceous shrubs really take over in disturbed bogs or bogs that are a little bit drier. And so one thing that we've hypothesized is that this really strong below ground response of the shrubs is one way that those ericaceous shrubs can kind of get a toehold to do a little bit better under warmer and drier conditions. We found that larch, Larix, the deciduous evergreen, also does a little bit better in terms of root growth with warming, but we didn't see any response of spruce, the sort of evergreen conifer. Dr. Camille Dufresne comes from a background in thinking about that partnership of root and fungi, mycorrhizae, and so she's been really focused on what are the fungi doing. So in a recent paper that Camille has submitted, she found that fungi are responding to warming as much as the roots are, and that they are staying sort of very shallow in the soil, which is interesting. So they're not necessarily diving deeper under drier conditions. They're sticking to the surface.
So my name's Natalie Griffith. I'm a scientist at Oak Ridge National Lab. I'm an aquatic ecologist and biogeochemist, so I study the elemental cycling of carbon, nitrogen, and phosphorus in aquatic systems, including wetlands and peatlands, like we are doing at SPRUCE. And so there's two types of water that we're looking at here. One is the pore water. So pore water is the water that is within the soils. We're also looking at stream water. And so the larger peatland itself, the entire peatland, has a stream that drains it. So first I'll talk about the pore water. The pore water, we're looking at the chemistry of that water at multiple depths into the peat. And so to do that, what we do is we have these PVC pipes, and so we drive it down into the peat soil all the way to the bottom. And at one spot within that, in this case, we have a 3-meter pipe, has some slots on it, very thin slots, and it allows water to come in to the pipe. And so we have 6 of those pipes, we call them piezometers, in each of our enclosures. And they have the screens, those little slots at 10 centimeters, 0 to 10 centimeters, then at 30 centimeters, at 50 centimeters, and then 1, 2, and 3 meters depth. And so we get a really nice samples from all the way at the very top of the peat, all the way to the very bottom to 3 meters. And then we look at the chemistry of that water. We look at things like the pH of the water, the specific conductivity of the water, and a variety of elements, things like from aluminum, iron, calcium, potassium. We look at the nutrients, things like ammonium, nitrate, phosphate. We also look at the carbon content of the water. And then with the stream water, so as I mentioned earlier, the entire peatland has a stream that drains it. And so in each of our enclosures in spruce, we've also essentially made a little mini peatland. To do that, what we did is we installed what we call a subsurface corral. It's made of interlocking sheet piles. We'd installed it around each of the enclosures in an octagon. We have one spot within that octagon that allows water to come out as a little mini stream, where we have 2 slotted pipes that allow water to drain out naturally. And so what we do is we measure the stream flow, the amount of water coming off of each of the enclosures. And then we have an automated sampler, and it automatically collects water, 75 liters or so. Every time that amount of water passes, it grabs a sample, puts it in a bucket, and then we collect that water once a week. Look at the same type of chemistry we do in pore water. And so what are we finding so far? We are finding that the chemistry is changing in spruce in response to the warming and elevated CO2. So first thing with the stream water, we're looking at 2 things there, the stream flow and also the chemistry of that water. So for the stream flow, we're finding that stream flow is decreasing with warming. And that's because the trees are using more water. And so we're also seeing that the chemistry of that water is changing. We're seeing that carbon concentrations are increasing, and we're starting to see some of the other elements that are changing in response to the warming. We're seeing similar responses in the pore water, but only at the really surface, the shallower depths. When you go down to the deeper depths, even though we're still warming those peats, we're not seeing a whole lot of response in the chemistry there. So at the spruce site, one of the things that I do is measure decomposition. So decomposition is the process where organic matter, how they decompose in the environment, so how they break down in the environment and how fast they break down in the environment. When the carbon is produced by vegetation, it also then breaks down in the environment, and peat bogs accumulate carbon because the decomposition process is slower than the rate at which the carbon is produced by the vegetation. Decomposition is slower in peat bogs for multiple reasons. One is it's an acidic environment, so that slows the decomposition rate. Another is that it's cold, usually, especially in the peatland we're studying, and even northern peatlands, so Minnesota and further north, cold, so that also influences the decomposition rate, makes it slower. And so we have been studying decomposition in this peatland to see how our warming and elevated CO2 treatments are going to affect decomposition rates. We're measuring multiple different litter types. And so we have the spruce tree is one of the dominant trees. So we're measuring decomposition rates of the spruce needles. We also have one of the dominant shrubs. It's called Labrador tea, so the leaves of Labrador tea. And then we also have 2 species of the sphagnum mosses that are growing in the peatland that we're measuring decomposition rates of. And then we're also measuring decomposition rates of 2 of the roots, both from the spruce trees and the Labrador tea shrubs. And so how do we measure decomposition? What we do is we collect those litter types from the fields, and then we measure it out, we weigh it, and we put it into a mesh bag. And so that mesh bag is then keeping the litter inside a bag, but also allows things like bacteria and fungi to come into the bag, colonize the litter, and help it break down. We place them into the peatland, and then we wait a year to 10 years. We come at multiple times, we pull bags out. So after a year, we pulled some of the bags out. We brought them back to the lab, and then we measured their weight again, their mass. And the rate at which they lose mass is decomposition. So more mass lost over time means it's decomposing faster. We also measure things like nitrogen, phosphorus content. So we're looking at other elements, carbon as well. So those 3 elements we're primarily focused on to see how those different elements, those important nutrients like nitrogen and phosphorus, also get lost as the material decomposes. We're finding so far there's not much an effect of warming on these initial phases of decomposition. We've analyzed data through 2 years so far. We're getting a hint of an effect of warming on the roots as they decompose. But what we really think is happening right now is that the chemistry of the litter is affecting the rate at which it's decomposing more so than temperature. And so as the material continues to decompose, we think that the temperature response is going to emerge over longer periods of time.
Hi, my name's Jeff Warren. I'm a plant physiologist at Oak Ridge National Lab, and we're interested in understanding how the treatments affect plant carbon uptake and plant water relations, plant water use. One of the key things we're focused on is plant carbon uptake through photosynthesis. Plant carbon uptake is dependent upon the carbon dioxide concentration in the air. So the elevated CO2 treatments at spruce are increasing the amount of sugars in the leaves. And the plants are able to use less photosynthetic machinery in order to take up the same amount of carbon. We're measuring water stress through a number of different mechanisms. One is directly measuring the water potential within the plants, and we're also measuring the rate of water movement through the plants. There's several different plant species at the site that we're tracking. What we're seeing is that the species seem to have different responses. The larch is able to maintain the stomata open and keep using water and keep taking up carbon through photosynthesis, whereas the spruce, in response to warming, are actually shutting their stomata. One of the ways we've been measuring that is to measure the plant hydraulics. How fast can water move through the plant? And this is related to the cell size in the xylem. And the larger the cell size, the greater the water movement. But as the spruce and larch develop new tissue each year, they are acclimating their hydraulic system to the new conditions. They're doing that in 2 ways. One is by potentially changing cell sizes and the growth rates of the xylem. The other is by adjusting the leaf area. So we have seen some shedding of leaves, particularly in the spruce. The next layer that we're dealing with are the shrubs. The shrubs are doing quite well in the warmer plots, and we're seeing greater shrub cover, and so the shrub density is increasing. Another component is the sphagnum. As a consequence of the greater water stress that we're seeing within the plots, a changing amount of shrub cover, production rates for sphagnum have dropped off dramatically.
Hello again. Thank you for taking the time to view the virtual tour of the Spruce experiment. More information on the Spruce project is available on the website shown on your screen. At the time of this video, the Spruce project has been running for 365 days a year over 5 full growing seasons. We anticipate that Spruce will operate for a full decade through the year 2025. We invite you to return to the Spruce Project website from time to time to learn about our new science results. We also expect that in-person visits to the Spruce site will be possible again in the future and hope to see you in person. Bye for now.